So hello everyone, here we are again, and we're about to continue with our story on protein metabolism and amino acid metabolism. So in the last video we saw how amino acids are catabolized in the scope of the removal of amino group, and we saw that ammonia is released, which we've mentioned to be a highly toxic molecule, and we're going to discuss a little bit about ammonia toxicity today, and we're going to see one very important metabolic pathway which actually serves to detoxify toxic ammonia and to convert it to totally non-toxic molecule urea, which can be safely excreted from the organism. So today we're going to talk about ureagenesis as well. So let's start. So we know that the excess amount of nitrogen which is formed during amino acid catabolism is excreted from the body. So we have already mentioned, okay, it's toxic. It has to be converted to something non-toxic, but nitrogen formed by amino acid catabolism can be excreted in 3 different forms depending on the organisms in which these processes take place. So it can be excreted in the form of unchanged ammonia. And this occurs in amphibians because they live in water, and the water content in the organism is so high and the dilution of ammonia is so high, so the toxicity drops down, so there is no need to make things more complicated than they are, and ammonia can be safely excreted from such organisms. But all other organisms are actually fighting the ammonia toxicity in a way that they are going to convert ammonia to something less toxic or to something completely non-toxic and then safely excreted in the environment. So ammonia can be converted to uric acid in birds and reptiles, but in mammals and most vertebrates, ammonia is converted to urea and prepared for safe excretion from the body. So we know that ammonia is toxic, and we mentioned several times that it's especially toxic for brain, for brain and nervous tissue. So it has to be metabolized to something non-toxic, and that non-toxic molecule is urea, which is excreted from the organism. So we say, if you remember our practicals, we say that urea is the final degradative product of amino acid nitrogen. Because of the importance of this metabolic pathway, ureagenesis is marked with different names and different synonyms: biosynthesis of urea, urea cycle, ornithine cycle, ureagenesis, so many names for one metabolic pathway. This pathway occurs in hepatocytes, and it has a cyclic form very similar to the Krebs cycle. This pathway is discovered in 1932 by 2 scientists, Hans Krebs, the famous one. This guy discovered famous Krebs cycle as well, and his laboratory associate Kurt Hanselite. So sometimes this pathway is marked as Krebs-Hanselite cycle as well. This metabolic pathway consists of 5 enzymatic reactions. First 2 enzymatic reactions occur in mitochondria. The rest of the 3 reactions occur in cytosol. Since this pathway is divided between 2 compartments, mitochondrial matrix and cytosol, we have to have 2 transport systems, which role is going to be to transport amino acid ornithine from cytosol to mitochondria, and amino acid citrulline from mitochondria to matrix. So this is one figure representing the overall urea cycle, and I really like it. It was taken from the Mark's clinical medical biochemistry. So let's get an overview of the urea cycle, and then we're gonna go through these reactions one by one in detail. So this is mitochondrial matrix. So we can see that there are 2 reactions followed by 3 reactions located in cytosol, and here we see transport systems for those 2 amino acids. So carbon dioxide, which is found in our cells as a result of the oxidative metabolism, is dissolved in water. So it's actually found in a form of bicarbonate ion, and this bicarbonate ion undergoes reaction with ammonium ion or ammonia. We discussed about this equilibrium in our previous video. So those 2 molecules react to form carbamoyl phosphate, and reaction is catalyzed by carbamoyl phosphate synthetase 1, and the energy for the formation of new chemical bond is provided by hydrolysis of 2 molecules of ATP to 2 molecules of ADP. So now we see that the first nitrogen atom originates from ammonia, which undergo these detox processes. In the reaction number 2, carbamoyl phosphate undergoes reaction with ornithine by ornithine transcarbamylase to form citrulline, and still we can follow up the fate of nitrogen atom from ammonia. So citrulline formed is then transported from mitochondrial matrix to cytosol, where it undergoes 3rd reaction with aspartate, and now here we see the involvement of the 2nd nitrogen atom in the molecule of urea, which originates from aspartate. In the reaction catalyzed by argininosuccinate synthetase, and for the formation of the new chemical bond, again we need an energy provided by the hydrolysis of ATP, but this time to AMP and pyrophosphate. So argininosuccinate undergoes hydrolysis by argininosuccinate lyase or argininosuccinase to form arginine, and here we have our 2 nitrogens and carbonyl atom, and we can see that fumarate exits this cycle. And finally, in the last 5th reaction, arginine is hydrolyzed and its guanidino group is removed to form urea and to release urea by the action of enzyme arginase. And we have the resynthesis of ornithine, which can be transported from cytosol to ornithine to mitochondrial matrix to join the new urea cycle. And of course, urea is excreted dominantly and predominantly in urine. So now we're gonna go step by step. From where do we get this ammonia? Of course, from blood. And if you remember, we mentioned that in blood there is only a small amount of ammonia, ranging from 30 to 60 micromoles concentration. But apart from free ammonia, which is transported through blood, excess nitrogen from amino acid catabolism is also transported in the form of amino acids, and you saw from the previous slide those are mostly alanine and glutamine, which are 2 amino acids capable of fixing and binding ammonia, and they're responsible for their safe transport of ammonia from tissue cells to the liver, where this ammonia is going to be released from those 2 amino acids undergoing degradative processes to release ammonia, which is going to enter the first reaction of urea cycle. So free ammonia or released ammonia from amino acids undergoes reaction number 1, which is synthesis of carbamoyl phosphate. So if you can see here from a figure, ammonia and bicarbonate ions, they tend to form carbamoyl phosphate, and we need the energy in the form of ATP. So this first reaction occurs in mitochondria, and apart from hepatocytes, this reaction is observed to take place in intestine as well. The reaction is catalyzed by enzyme carbamoyl phosphate synthetase 1, and it requires coenzyme biotin for its action, as well as magnesium ions as activators. So here we have the usage of 2 ATP molecules hydrolyzed to 2 ADP molecules and inorganic phosphate. So the allosteric activator of this enzyme is N-acetyl L-glutamate, and this reaction, this very first reaction of urea cycle, is actually the major regulatory reaction of the pathway. Regarding the fact that we have mentioned carbamoyl phosphate synthetase 1, obviously that there is more than one, and yes, there is carbamoyl phosphate synthetase 2 isoenzyme, which is found in cytosol, and this one is involved in a absolutely different pathway, which is synthesis of pyrimidine nucleotides, where carbamoyl phosphate is synthesized from glutamine in order to be incorporated in the pyrimidine ring, in order to synthesize orotate and so on and so on. But this is going to be the topic for one of the upcoming videos. So anyway, after the reaction number 1, carbamoyl phosphate undergoes reaction number 2, and that is the reaction with ornithine, thus forming citrulline molecule. So this is the second reaction taking place in mitochondria, and the energy for the formation of new chemical bond this time is provided by carbamoyl phosphate itself, because it's a high energy compound. Reaction is catalyzed by ornithine transcarbamylase, or OTC in short. Maybe this is the moment to mention that OTC deficiency, if you can see, tends to the accumulation of carbamoyl phosphate. And I have mentioned previously that carbamoyl phosphate is involved in the pyrimidine biosynthesis. So accumulation of carbamoyl phosphate yields the enhancement in the biosynthesis of pyrimidines, thus increasing the concentration of the first pyrimidine nucleotide, which is, or nitrogen base, which is orotate, which is going to be excreted in urine. So actually, in OTC deficiency, no ill effects are observed. Citrulline, which is the product of this second reaction, is now transported from mitochondrial matrix to cytosol to enable the molecule to undergo reaction number 3. So citrulline undergoes reaction with aspartate to form argininosuccinate. So this is the first reaction, the third one, but the first taking place in cytosol. So here we have the entrance of aspartate into the cycle. The reaction is catalyzed by enzyme argininosuccinate synthetase, and this one requires magnesium ions as activators as well. And again, for the synthesis of new chemical bond, energy is required, and this energy this time is provided again by ATP molecule, but this time upon the pyrophosphate cleavage to AMP and pyrophosphate group. Argininosuccinate undergoes hydrolysis and degradation to arginine and fumarate. This reaction number 4 occurs in cytosol as well. And in this reaction, we see that fumarate exits the cycle. Fumarate can now be used either for regeneration of aspartate or for glucose synthesis. This reaction is catalyzed by enzyme argininosuccinase or argininosuccinate liase. Those are two synonyms found in different sources, textbooks, and both are correct. And the final reaction, the last one, the fifth reaction, is hydrolysis of arginine to urea and ornithine, which closes the cycle. This fifth reaction occurs in cytosol, and it yields the formation of ornithine, which is going to be transported now from cytosol to mitochondrial matrix where it can accept the new molecule of carbamoyl phosphate. The reaction is catalyzed by enzyme 1-hydrolase, which is called Ureagenesis, and this one is found only in hepatocytes. Urea, which is actually released, if you can see, from guanidino group of arginine, is now ready for excretion. At our practical exercises, we discussed in details how urea can be excreted by kidneys, gastrointestinal tract, and to a lesser extent by skin. And now I'm not going to go into these details all over again. We're going to get back to our urea cycle. So let's analyze the origin of atoms in urea molecule. So we saw from the overall pathway at the very beginning that the first nitrogen actually originates from ammonia or ammonium ion, which enters the cycle. The second nitrogen originates from aspartate. If you remember, it was a blue shaded amino group. And carbonyl group, CO group, originates from dissolved bicarbonate ion in mitochondrial matrix or dissolved carbon dioxide. Now there is a question regarding energy yield. So if you calculated carefully from our previous 5 reactions, so for the biosynthesis of one molecule of urea, 4 high energy bonds from 3 molecules of ATP are required and used. So one can conclude that this is actually an endergonic process, but we always say that the urea cycle belongs to the group of catabolic processes. So obviously that there are some other alternative connective pathways from which ATP is going to be synthesized and used for those synthesis of urea molecules. And one of those connective pathways is definitely regeneration of aspartate molecule. So we also saw that fumarate molecule leaves the urea cycle, and fumarate is a molecule which is one of the intermediates of now our very well-known pathway Krebs cycle. So in Krebs cycle, fumarate is going to be converted to L-malate by the action of enzyme fumarase, and then L-malate is going to be oxidized to oxaloacetate by malate dehydrogenase. This is also the reaction of Krebs cycle, and you know that malate dehydrogenase requires NAD coenzyme, and reduced NADH is going to be formed, which is going to provide 3 ATPs in electron transport system. Oxaloacetate now can be converted to either glucose in gluconeogenic pathway, or it can be converted to aspartate in the reaction catalyzed by aspartate transaminase. And if you know from the previous slide, transaminase is always connected and followed by deamination processes. So one other process leans on transamination, and that is the deamination of L-glutamate to alpha-ketoglutarate, which again enters the Krebs cycle, and again we have the formation of NADH coenzymes to provide 3 ATPs. So when we calculate all these, the total number of ATPs which can be released from this actually overcomes the number of ATPs which were required for the biosynthesis of urea molecules. So the net energy yield is actually positive, and urea cycle is considered to be a catabolic energy yielding pathway. It's not going to be a huge amount of energy, but still it's energy yielding pathway. What about the regulation of urea cycle? So before we move on to this, it is important to emphasize that there is a really vast capacity of this pathway to convert ammonia to urea and thus prevent its toxicity. the first way to regulate this process is definitely the availability of the substrate, and this is the common regulatory pathway when we are talking about those disposal pathways which actually yields the formation of a molecule which is going to be ready for excretion from the organism. So what is the mechanism of this regulatory pathway? The higher the concentration of ammonia, the more intense urea cycle. And the second is the one that we have mentioned in the reaction number 1, and that is allosteric activation by N-acetyl N-glutamate NAG. So it activates carbamoyl phosphate synthetase, which we've mentioned that it's the main regulatory site and the main regulatory enzyme of the pathway. So NAG does not have any other known functions but to activate this enzyme. NAG is synthesized from acetyl coenzyme A and glutamate by the action of NAG synthetase, and arginine is the one which actually activates this enzyme, thus forming more N-acetyl L-glutamate. So what does this mean? And how does this function? So if from any reason you have an intensified or enhanced amino acid metabolism, you know that from transamination processes you're going to have an accumulation of L-glutamate. Then L-glutamate, as one of the reactants, is going to promote and shift the equilibrium towards the synthesis of N-acetyl L-glutamate. And the increase in NAG concentration is going to enhance and intensify urea cycle, thus directing the transformation of ammonia released from amino acid catabolism to be converted to urea and to increase the excretion of excess amount of urea and amino acid nitrogen from the body. So cycle activation is also achieved by protein-rich diet or during prolonged fasting. During prolonged fasting, you have to take into account that you have actually two pathways. We said in the previous slide that during fasting, muscle proteins are going to be depleted to amino acids, and then amino acids are going to undergo two pathways. So carbon atoms are going to be directed to form glucose, and nitrogen is going to be redirected to form urea. So, if there is an inherited deficiency of any of the cycle enzymes, the result is going to be accumulation of toxic ammonia, which is especially toxic for nervous system and brain. So, the cycle enzymes deficit is going to develop mental retardation and lethargy. And the absolute deficit, absence of any of these enzymes, it's gonna end up as a lethal effect soon after birth. So let's explain these symptoms from the biochem point of view. So, if from any reason, so for example you have deficiency of any of these enzymes, so ammonia is not going to be converted to urea efficiently, so the result is going to be the accumulation of ammonia, which means that the concentration of ammonia increases. Now we are going to use our chemistry knowledge and the shifts of equilibrium and Le Chatelier principle and the knowledge from our previous slide. So, we mentioned previously that one of the important processes in amino acid catabolism and amino acid nitrogen metabolism is deamination. And the reaction, if you remember, goes L-glutamate to alpha-ketoglutarate plus ammonia. So if you're following this reaction and ammonia is increased, that means that according to Le Chatelier, Increasing one of the products of the reaction shifts the equilibrium towards the reactant, which means that the concentration of L-glutamate is going to be increased and alpha-ketoglutarate is going to be used. So, alpha-ketoglutarate is an intermediate of the Krebs cycle. So, if you have an accumulation of ammonia, you need more alpha-ketoglutarate to neutralize it to L-glutamate, which means that you're going to use those reserves from Krebs cycle. So alpha-ketoglutarate from Krebs cycle is now being consumed. After a certain amount of time, and it's going to happen very quickly, you're going to have, as a result, you're going to have a significantly impaired normal Krebs. And indirectly, you're going to have an impaired flow of all other metabolic pathways that are somehow connected to Krebs. And those are almost all of them. So you're going to have a disturbed and disrupted carbohydrate and lipid metabolisms as well. Let's move on. Once L-glutamate is formed, so we have ammonia using up alpha-ketoglutarate, forming L-glutamate. But still, we have the continuous accumulation of ammonia. So we have L-glutamate and ammonia. We mentioned in acid-base equilibrium, if you remember. If not, let me tell you all over again. L-glutamate has a carboxyl group in the side chain. So if there is a lot of ammonia and you have a carboxyl group, they tend to react to form an amide group. And if you have an amide group in a side chain, then you have an amino acid glutamine. So L-glutamate formed plus excess ammonia tends to produce glutamine. And as a result, you're going to have accumulation of glutamine in a cell and decrease of L-glutamate in a cell. What is the consequence of the decrease of concentration of L-glutamate? That is insufficient synthesis of GAB, gamma-aminobutyric acid, which is neurotransmitter on one side. On the other side, you have decreased L-glutamate, which is neurotransmitter itself. And obviously, that is going to be directly reflected negatively on the function of central nervous system. Let's get back to our Krebs cycle. We have an impaired flow of Krebs cycle, and it disrupts the flow of glucose metabolism or catabolism. I'm mentioning that because we know that glucose is a major energy source for brain. So even from that point of view, it contributes even more to the negative effects of accumulation of ammonia. And now we have an increase in glutamine. So after all, we have an increased concentration of glutamine, which can cause the brain swelling. Why? Because glutamine, together with ammonia, those are all osmotically active compounds and molecules. So you're going to have a disrupted osmotic balance in astrocytes. Also, the membrane permeability is going to be changed, actually increased. And glutamine is going to cause the opening of mitochondrial pores, which is going to lead to cell death. And explaining the lethargy as a clinical effect, it's caused by the loss and deficiency in L-glutamate, which is actually an excitatory neurotransmitter. Most oftenly, in most of the patients, OTC deficiency is one that is going to be observed. And the major problem in this is how to reduce effects of excessive blood ammonia on the nervous system. And of course, scientists and health professionals, they provided some possible and suggested and proposed some possible solutions. Obviously, early diagnosis of the problem is extremely important. And then therapy with certain compounds that can aid the nitrogen removal, and I'm going to illustrate that to you later on. And of course, low protein diet is of essential importance because you're gonna decrease the flow of amino acids to organisms, so less amino acids are gonna undergo catabolism and ammonia formation. So if you want to analyze the effects of this enzyme deficiency, you have to figure it out and solve the problem depending on where is the origin of the problem. So if there is a defect of the enzyme activity after argininosuccinate is synthesized, then the problem can be solved or at least reduced by massive arginine supplementation. If you remember, arginine in urea cycle actually is hydrolyzed to urea and ornithine. So if you have arginine supplementation, it's going to be hydrolyzed to increase the ornithine production. Ornithine is going to be, of course, transported to mitochondrial matrix, so it's going to be ready to increase the uptake of carbamoyl phosphate, and the increased uptake of carbamoyl phosphate is going to increase the usage of ammonia and carbon dioxide and bicarbonate ion to form and synthesize carbamoyl phosphate. But if there is an enzyme defect before argininosuccinate synthesis, then supplementation with arginine is completely useless. But in that case, you can use medicines that form conjugates with amino acids. And those conjugates cannot undergo further catabolism, so they're going to be excreted. And actually the point is that now those amino acids have to be recycled, so ammonia is consumed for the resynthesis of those amino acids. I'm going to mention two examples, two compounds. The first one is benzoic acid, which actually reacts with amino acid glycine to form hippuric acid. And this is also the reaction which takes place in detox mechanisms in the liver, but now we're not going to discuss about that. And hippuric acid is then excreted from the body. So due to the fact that we are actually using glycine for the purpose, glycine has to be resynthesized, and we will see in the next slide that it's going to be resynthesized from serine. And for any reasons, for glycine and serine, you will have to use ammonia, and thus you're going to decrease its accumulation in a cell. In the other way, you have phenylbutyrate, or phenylacetate. So actually it forms conjugates with glutamine, and those conjugates are excreted. So you're targeting actually glutamine, which is the final molecule accumulated in the body. And of course, because ureagenesis is exclusive for hepatocytes, actually the patients with these defects, enzyme defects, are excellent candidates for gene therapy, which is especially promising because you don't have a wide range of different cell types and tissues to fight with, so you have to target only hepatocytes and find an adequate gene therapy solution. So actually, my dear students and my dear colleagues and listeners, that was all regarding the amino acid nitrogen metabolism. In our next video, I'm gonna focus on the fate of carbon atoms from amino acid, which is gonna be rather complex. But anyway, until then, stay healthy, stay safe, and bye-bye.